Yeah, so you can think of an exosome like a packet of information or a message that one cell is sending to another cell. It's protected by a lipid bilayer. It's kind of like cellular mail, right? And so we want to capture the mail from this cell in transit. And so that kind of brings you to thinking, well, what cell do we want to steal the mail or the messages from and why, right? And that's really the kind of mindset here.
Ah, totally understand. I said somebody explain it to me, something like an Amazon delivery. And then,
The messenger.
yeah, so we want to say, I think it was something like the delivery truck was the stem cell, and then inside we have the packages, and the packages would be a bit more the exosomes because that's what's inside what needs to be delivered. Is that more or less an analogy that's
Yeah, I often use the analogy of like an IKEA package, right? Because you have all the building blocks, they give you the tools, they give you the instructions, it's all together in one neat package. And that's often the case, right? It's not one single thing that we need, but this mixture that all is meant to interwork together. From my point of view, as a stem cell scientist, I was really impatient with wanting to see this kind of technology make it to practical use. And unfortunately, most people don't have a stem cell laboratory. They don't have the liquid nitrogen tank to store them. They don't have all the things you need to really grow them, culture them, benefit from them. It's not an off-the-shelf thing. I mean, people have tried and there's methodologies there, but it's very difficult for the average person to benefit from stem cells in their form because we don't all have a laboratory environment. Whereas the exosome, not being alive, lipid encapsulated, if it's treated the right way, can be stabilized and that can be put on the shelf. And so it allows you to grab arguably the best part of stem cell technology and put it on the shelf.
I had a discussion before about exosomes versus stem cells, and it was a discussion around regenerative medicine in terms of pain and restoring cartilage and things like that. So I understood the message that I got away from that was, okay, well, stem cells are much more expensive and exosomes are more accessible in terms of finances. And it kind of does the same thing in terms of that, you say knee pain. Would that be correct? Or do you think that there's more nuance to that?
I mean, the analogy I often use is you can think of the stem cell as sort of the fire department, and the exosomes are the firefighters that are going out and dealing with the problem. The fire departments of our body, our stem cells, are getting exhausted as we age. They break down, they're not releasing what they used to release. And so I think this is where this has become exciting because we can borrow the healing capacity from very young stem cells and bring it to our aged cells. And again, it's not alive, it's not dividing. You don't need to culture stem cells yourself to get that benefit. And while there's an argument, I think, to be had where people have tried to inject stem cells or inject the factory that's producing the exosomes, for most of us, there's a lot of legal limitations, regulatory limitations, there's safety concerns. This is much easier. And of course, we're still limited. Like there's nothing that's a legally approved injectable exosome in the United States. But certainly there's a lot that we can benefit here by grabbing these biomolecules from young stem cells. In the United States, there's no legal injectable exosome products on the market. Anything injectable by default is considered a drug. And you have to ask yourself, like, what is your goal? Because there are certain things that you could make an argument for why you might want to inject. But here we're talking specifically about the skin. And I think there's tons of benefits that you can get topically that don't require injection. And that has to do with not only the payload of the exosomes, but they are nano size. They're very small. So even like a sweat gland, hair follicle, these are thousands of times larger in diameter than the average diameter of an exosome. So it's a bit like throwing a marble down a well when you think about these exosomes. I think a lot of people just don't realize that. They're thinking that they can't possibly get in without injecting them, and that's simply not the case. Yeah, I actually I've grown all different kinds of stem cells from different sources. Initially, I was using adipose tissue because people are getting liposuction done every day and you could get a hold of fat very easily. But of course, people who are doing liposuction are not necessarily the healthiest or the youngest, right? Same with bone marrow. Whereas a lot of folks are saving the umbilical cord and a lot of folks are not. They're throwing it away, right? And so inside the umbilical cord is something called Wharton's jelly, and you can isolate some mesenchymal stem cells from that and in an ethical way, in an almost surgical way that's pretty pristine. And so that's the cell source of choice, in my opinion. It's something that has got no age to it, right? So if you're looking at a mesenchymal stem cell source, you want the youngest possible that's pristine and hasn't had any environmental impacts. Of course, it has to be tested to be free of all kind of communicable diseases, and there's multiple tests throughout to make sure there's no microbial contamination, things like that. But arguably, those cells they have the blueprint for perfect
Yes.
healing, right? The healing we had when we were very young. And so that's really the goal to get to that.
So you're saying these are coming from umbilical cords and they're harvested or grown more, or they just come as there is? How does that work?
It's a small portion of the umbilical cord. It's not taking everything. Yeah, so you got to isolate them carefully from the umbilical cord. You have to run them through a variety of tests. So look at the surface markers, look at what they're expressing, make sure that you actually have the right population of cells. But I actually published a paper, I co-authored a paper last year that looked at all of the protein work that had been associated with mesenchymal stem cells. And so we did a bit of a meta-analysis on all the peer-reviewed published data on mass spectrometry data from different stem cell types. And we showed that there was a lot of overlap, as you would expect, between an umbilical mesenchymal stem cell, a bone marrow source, an adipose source. But in the umbilical arm, there is a couple thousand proteins that were unique to the umbilical versus adipose and bone marrow. And we believe that it's in that bucket that the
Mhm.
difference lies, right? And so we experience it as aging, but the reality is it's a shift over time in gene expression.
Dr. Suzanne Faree tested this with us, and she said her skin, one of the things, and I think she's using it maybe 4 to 6 weeks. We haven't, it is not out there, and Jordan can talk more about people who've used exosomes separately from Vita, but she talked about how refreshed. She said her skin just had this refresh, the texture was better. I've had someone tell me that a couple of the other people about their pores. She said she had always had big pores and said since she's been using Vita, she's just felt the refinement of the pores and just much tighter.
It kind of is a repeat of what we saw with stem cells. So at first, stem cells came out. People, I think, started very afraid. There was a lot of stem cell research that was banned in America under George Bush. And then 2008, 2009, Obama overturned that. And all of a sudden, there was a Wild West. People are injecting stem cells. The FDA is saying, no, 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 you can't do that. But basically, there's a whole shift from fear to excitement. And then you start to see companies all of a sudden slap stem cell on every single type of product out there. All of a sudden, there's apple stem cells and plant stem cells and all this stuff that really
Mhm.
doesn't exist in the literature. They're not being studied in the same way. And so for our human biology, I would argue you want a human exosome product. And of course, it needs to be made safely and effectively. One of the things there is I wouldn't want to use a product where the cells were exposed to animal components. And that's a common thing if you're growing cells in a laboratory. They often use something called fetal bovine serum, and that can have mad cow disease and prion-based things in them. So there's no animal-derived components used in the processing. I think that was one of the first things I told Debbie and Terry was that's the safety thing to really look out for. There are other things that people will bring up, but I think that's the number one safety concern that should be easy to, you can ask a company. Some parts of the world simply do not allow a cell-derived protein or anything to go into a skincare product. And so in those markets, you will find predominantly plant or bacterial exosomes, and they might have some internal data that they've made to try to support that. In my experience, it just doesn't compare to the human form. And it's like if you went into the hospital and you needed a blood transfusion, they wouldn't come up to you and say, oh, well, can we give you some tree sap? We're out of blood. It's the closest we've got. You probably wouldn't be a fan of that, right?
I have always thought about the skin as an organ, your largest organ. So I've treated it that way. And I think about what I eat. All of this, I think about sleep, eat, must have enough rest. I know my skin looks the worst when I don't consistently. The diet, the kind of things, no seed oils. Exercise because that's so important for the oxygen and keep the blood flow for your skin. I've always been very careful, even though it's only been 5 years, a little over 5 years that I discovered copper peptides. I always was careful about not putting junk on my face. So I would go in and I would have these apps of the things to avoid. And I would get people would look at me, I would be in the pharmacy or whatever and reading everything, all the sunscreens. So I think a big portion is being careful not to put toxic, bad things on my skin. And I do that with my children and grandchildren too. I mean, I've snatched things out of their hands because they were bad. So I think that's a big, and then what's so wonderful is 5 years ago when I was worried about my crepey skin and went to Dr. Faree and said, okay, what have you got? You've done all these wonderful things for my muscles and my other things. What can we do for the crepey skin? And that's when she said GHK-Cu. And after 5 years, it dramatically changed. I mean, it's better than it was 10 years ago. So
Wow. Yeah.
maybe not 20 years, but you can make up some time if you have the right products and you don't put bad things on your skin.